The overall goal of the following experiment is to assay toxicity of the beta-amyloid peptide in an in vivo model system. The reproducibility and relative simplicity of this assay allows rapid testing of drugs, environmental conditions, or genetic modifications on beta-amyloid toxicity. A key factor in developing this assay is the generation of transgenic C. elegans worms with muscle-specific, temperature-inducible expression of the beta-amyloid peptide. This temperature-sensitive expression is engineered by constructing a transgene that contains an abnormally long 3' untranslated region, making transgene mRNA a target of the mRNA surveillance. Expression of this transgene is therefore poor in wild-type worms with a functional mRNA surveillance system due to degradation of the transgene mRNA. If this transgene is introduced into a genetic background that contains a mutation in an essential component of the mRNA surveillance system, transgene expression will be significantly higher. If the genetic background contains a temperature-sensitive mutation in the mRNA surveillance system, then propagation of this transgenic strain at the non-permissive temperature will block transgene mRNA degradation and result in high transgene expression. In transgenic worms employing this system to regulate body wall muscle expression of the beta-amyloid peptide, temperature upshift from 16 degrees Celsius to 25 degrees Celsius results in high accumulation of the beta-amyloid peptide, dysfunction of muscle cells, and paralysis of the worms. Measurement of the time it takes a population of these transgenic worms to become paralyzed after temperature upshift is a sensitive assay for beta-amyloid toxicity. This is achieved by making fresh nematode growth media plates for the paralysis assay.